Let's get going. My name is Ian Horvath. I am president of the of the Portland Urban Beekeepers. And tonight we have Ramesh Sagili from OSU coming to talk to us about honeybee nutrition. So let me run through some of our housekeeping things and then we'll hand it over to Ramesh. Our little shameless plug, if you're not a member, we do encourage you guys to join and help support our mission. So some of the benefits that you get with that are you get put on the swarm list. We've had 80 plus in the last 2-ish months, something like that. So it's really ramping up and I feel like we're just at the beginning of the good weather, so that will probably expand. We have our Bee Buddy program. Last I checked, added something the other day. We're up to 50 now mentors and learners, and that's a really great way to connect with people in your area. We have the bee days at the apiary, so we try to do those every other weekend out there. And as the weather gets better, we'll get better about going out there. We also have our library, our rental equipment. You can post on our classifieds on the website. And then lastly, the extra funds every year do go to funding bee research. So last year we donated 20,000 to Ramesh and Andoni down at OSU, which was really awesome. So hopefully get close to that again this year. And again, it's all that for just $25 a month or $25 a year. The Bee Buddy program, like I said, we're up to about 50-ish people on here. And it's a really great way now that we're still on Zoom to meet people in your local area. So you can see we have mentors and learners, and you just reach out to somebody once you get put onto the map and see who's around you and connect. And it's great for people who want to learn together to partner up and then go to a mentor's place or have a mentor come over to your apiary and just talk to you guys. And you can see a little bit about them and everything once you click on their little icon. So I've said it a few times, but I'll say it again just because we're still in swarm season, that we did upgrade the swarm software. So if you have not re-signed up in the last 60 or 90 days, 3 months, you need to sign up again for the swarm software. So we have instructions on how to do that, and I can go over that with you if you email Jana or myself. But it's really easy. You're just going to go to the website. You get a registration code, and that's what would link you to the Portland Urban Beekeepers. You just fill out your information and then submit it, and then you're on the swarm list. One of the important things is the range in here, so you can specify how far you're willing to travel, and that will also move you higher or lower in the list. So if a swarm is closer to you and you're higher up on the list, then you will get a call before somebody else. So, all right, Bee School is nearly done, but all of them are recorded, and we still have one more. But Bee School is really great for the beginning beekeeper. It's got a ton of information. Susie, who is on the board, she has done this now for 2 or 3 years, and this might be the 3rd year, and she's awesome at it. It's tons and tons of information. We use Dewey's book, the Honeybee Biology and Beekeeping book, which is really great. And then they also do go out to the apiary and kind of have their own bee school day out there with Dewey, which is really cool to meet him in person. So it's 4 classes for $45. We've done 3 of the classes so far. They are recorded though, so you can watch all of them in the past. And then we also have one coming up, whatever day that is, next week sometime, maybe Friday or something. So really interesting stuff. Actually, this is a great time to be looking at your fall and winter management as well. So I know we're just feel like we're getting started with bees, but we need to always be looking forward at planning ahead. So, all right, we had a bee day at the apiary on Sunday, I believe, which was awesome. You guys did inspections out there. And then we also have one coming up in May. So this is our agenda for the May 15th one, is going to be bee inspections again on the Langstroth hives. We're going to be checking on the results of the split. So we did a split last Sunday. We're requeening 1 hive, adding supers possibly, depending if there's needed, and then hive inspection on the top bar hive. So that's really fun. It is out at Green Anchors and from 10 to noon on the 15th. Here's a little map that you should park in the park over here, and then walk around and then come all the way down to this big white open structure. There'll be a lot of other bee geeks in their beekeeping suits there, and then our hives are just spread out over here. So a lot of fun. I will drop this address in the chat for anyone who wants it, but it's up in the St. John's area. So All right. So if you joined earlier, you heard me speaking a little bit to Ramesh about there's been some stirrings on possibly going back into person. The board has talked about it a few times, and we want to now take the pulse of the community and see how many people would want to go back and how often they would want to go back and where we would want to go back as far as location and everything like that. So I'm going to be putting together a short little survey. Hopefully it's just 5 or so questions. If you want your voice heard, then take the survey. If you don't, then don't complain about the decision we make. So, all right. And lastly, we just want to go over the upcoming speakers. So we have Ramesh tonight. In June, we have Dewey talking about queens and getting to know your queens and bees better. In July, we have Cassie talking about the Asian giant hornet. So she's from the Washington agriculture, which is really interesting up in the northern Washington area. And then in August, we have Randy Oliver coming to talk about his OAV research, some of the stuff he's just completed. In September, we have a panel on winterization, which is going to be really cool. So we'll hear from 3 different people and how they each approach winterizing hives, which I think is great to get a diversity of opinions there. And then in October, we have Matthew Shepard from the Xerxes Society. So this is really exciting. He's going to be talking about bringing back pollinators, bees, and native bees, and how we all incorporate together. So without further ado, I will hand it over to you, Ramesh, and stop sharing my screen.
Thanks, Ian, and thank you all for being here. Yeah, I saw my department head going back. I came back to office from home again, so that my dog and my younger one, the teenager, doesn't make much noise there. So yeah, I was saying, like, I wish our beekeepers meet at 5 o'clock so that I can give my talk and go home rather than coming back again at 7. But I know this time works for everyone. I really love it. I'm not complaining. So, okay, I will start sharing the screen here. Can you see, Ian, my screen?
Yes, we can. We can see your PowerPoint now. Yep.
Okay. See a full screen now?
Yep, looks great.
Perfect. Okay. So do I have like 45 minutes or an hour? You know, I'm notorious. I can go over time easily. So I'll respect your time. Is it 45 minutes, an hour max?
Sure. Yeah, let's keep it 45 minutes, an hour, and then take questions at the end. That'd be great.
Okay, perfect. Yeah. So I know I sneaked in EFB as well. If you see in the title here, I know there is European foul brood problem sometimes in spring. So I thought I'll just ask you some questions and go over a small project that we started last year on looking at EFB in commercial beekeeper hives. So yeah, I'll see if I have time, I'll go over EFB as well, but let's focus mostly on honeybee nutrition, which is a topic. You know, nutrition is not just important now, but it's throughout the year, right? It's really critical. So I'll go over some of the research we did, and maybe some of the information might be useful for just before I start, Ian, like, are there several new beekeepers as well that are just starting this year in your group?
Yeah, I think we have a good mix of new beekeepers and then 2 or 3 years and maybe a little more seasoned as well. But I'd say mainly new beekeepers are joining the meetings right now. Yeah.
Okay, perfect. Yeah, the reason I ask is so that I don't go too much deep and try to confuse or overwhelm them. So I'll keep it more basic as well sometimes. Okay, before I start, I know you can either scan this or if you have, I don't know if some of you have used this, slido.com. I just keep that so that you guys don't sleep after your dinner. So I may have a couple of questions there that you may have be. So yeah, it's pure volunteer. Yeah, if you want, you can scan that, the one you see now, the code there, or you can go to slido.com, and then when the time comes, you have to put that number in, 969422. Okay, so yeah, before I start, I have to really thank all these people, these wonderful people, and many of you know several of them here. Carolyn, Hannah, Priya, who was a postdoc, she just moved to Mississippi State University. She got a assistant professor job there. And then Jen Larson, who is the Oregon Master Beekeeper Coordinator, which is a popular program that we run here as well. Ellen and some others. So yeah, these people, I mean, several of them have left, but again, everyone that's shown here is really instrumental for this program for the last 12 or 13 years now. And of course, Portland Urban has been supporting us with funding for a long time as well. So we really acknowledge your support. Without passionate support from beekeepers like you, research is not possible. So here is what I'm gonna do in the next 45 minutes, give you some basics of bee nutrition, especially for the new beekeepers that I was asking. You need to understand why it is so critical. And then what are the nutritional challenges that our bees face as both backyard beekeepers or commercial beekeepers. And then I'll go over some of the ideas of how to improve habitat for bees and what are the challenges associated with this. And if I have some time at the end, I'll try to sneak in some European fall brood slides as well. Okay, this adage probably most of you have heard. We all talk about you are what you eat, and that's what our bees are also, right? They need a good diet as well. And nutrition is the first line of defense. I don't have to say that. It's pretty much understood. If you have a better immune system, and for better immune system, you need better nutrition. And then probably you can fend off some of those problems coming from stressors, like anything from pesticide exposure to a virus or rheumite feeding on your abdominal fat bodies in case of bees. So nutrition again can protect our bees to some degree for sure. So I think we shouldn't ignore nutrition. And several studies have shown optimal nutrition will boost the immune system, as I said before, and also helps detoxify some of the toxins that they bring in. So usually I want you to think nutrition is not simple, like just, oh, I have nectar or honey, and then my bees are gonna eat the carbohydrates, and then we have pollen. It's not that simple, right? Our honeybees are so social that we have a complexity of this nutritional angle that you want to think about. So you want to think of three different levels here. There is a colony level nutrition. That means it's a bigger picture, right? And then you have adult nutrition. Again, foragers will have a different needs for food, right? And your nurse bees are eating pollen. So foragers that collect pollen, they never eat that pollen directly. They just deposit it, and the nurse bees, which are about 7 to 14 days old, that's what we categorize them as nurse bees. Those are the ones eating most of the pollen and growing those hypopharyngeal glands and mandibular glands. And that's where the food comes from, right? And these are called exocrine glands. That means they are opening outside. So those head glands here and the mandibular glands, they're connected to the mouth. And that's how all the brood food, that white stuff that you see on your larval cells, that's being produced by those glands. And those glands really need that pollen to be there. So nurse bees are the ones that are eating pollen, not your foragers. Foragers are old now, right? They are probably 21 days plus, and they survive maybe in a busy season like now. Foragers may survive maybe 2 weeks max. So they are at the end of their lifespan. So they don't need that much protein. They do need some. So they beg these nurse bees when they come in, and the nurse bees will also regurgitate some protein for those older foragers as well. But again, foragers' job is to just bring in stuff. They will feed on the nectar and honey because they need a lot of carbs, but they don't need that much protein. So that's what I say here. Adult nutrition is very different, again, depending on the age group as well. And then larval nutrition is very different, right? I mean, if you think about some cockroaches, or maybe think about some of these insects that have nymphal phases, like grasshopper, right? So grasshopper, you'll see the baby grasshopper is also similar to, I mean, the wings may be smaller and all those different morphology will be different, but they are still eating the same diet, right? Grasshopper will eat those leaves or whatever the plant species they are on, and your younger nymphs will also feed on they don't have a different diet. The mother is not feeding something very different there, right? Or they're on their own. So, but in your honeybees, being this very complex social structure, and this is one of the most advanced insects we're talking about, right? So they have evolved for these millions of years to develop this nutritional differences as well. So larval nutrition is all coming from those glands, right? It's like a cow's milk if you're comparing to a mammal. So larval nutrition is very different. They can't directly eat everything. There may be a small amount of pollen at the later stages of larva, a couple of grains that are getting in, but otherwise the total diet is from those two glands they talked about. So again, as a beekeeper, you may say, oh, why are you telling us all this? I think if you understand this, maybe then at least you'll understand what you're feeding your bees. And I think it does make sense at the end. So bear with me when I go into a little more basics here. So this is what you have to think about a colony. It's not just that pollen is coming in, so our bees are fine, but you have to look at colony level nutrition, there is adult nutrition, and then there is larval nutrition as well. So this is a more simplistic explanation of what you see in your hives, right? So on the left of this line, the black line you see, I have put some macronutrients there. So macronutrients means your bees need them in large quantities, right? That's what macro means. So you have carbohydrates and proteins. Those are the two important ones. So protein is only pollen for them, right? We can eat a lot of things from beans or to chicken or some other meat for your protein. But unfortunately, the only source of protein for bees is pollen. And that's where the protein comes from. And carbohydrates, of course, the nectar, or if it's processed into honey, that's what they are eating for carbohydrates. So they need a lot of honey during their development and their growth. But pollen, people have shown, at least there are a couple of studies that have shown, they don't store more than a kilogram at a point of time. So that's why when there is a lot of rainy days in between, they run out of pollen. And that's why we see colonies struggle to grow, because they don't store pollen as much as they do honey stores. So again, I'll not go too deep here, but just to give you a sense, pollen, like that's why you see smaller amounts when compared to honey in your colonies. And then on your right is the micronutrients. So those are smaller amounts you need, right? The vitamins, minerals, or lipids. I gave you an example here, which is sterols. So we need those, they're very critical. So unfortunately, even though honeybees have been studied for 200 years plus in a modern world, very properly with experiments, very meticulous experiments, we still don't know the right side here. The micronutrient understanding is literally nil, but we know much about this macronutrient. So this is where our lab is focusing on a lot. So we do a lot of work on honeybee health, including mites, nosema, EFB, and other bacterial diseases, but nutrition is another prime area that we have been focusing on the last 6 or 7 years, trying to understand the basics. What exactly bees need in terms of sterols. So most of the work was done in 80s, and after that, they dropped the ball. So there is not many papers. If you try to look for some scientific publications, peer-reviewed journals, most of the work was in the late 80s or early 90s. And after that, there is not much understanding of micronutrients. So I'll show you some studies that we have done and published in the last few years as well. So those are more basic, trying to understand what the bees need. So I'll start with pollen, because that's what we have been focusing more on. So pollen is, as I said, is the protein source, and it could vary between pollens. Don't think you may sit next to your hive and see some yellow pollen coming, some brick red pollen coming in. Those pollens are very different in their composition. They don't think all pollens are made equal. They may vary a lot in terms of protein that I'm talking about here. They could range anywhere between 10 to 40%. And then they may have very much differences in the sterols. There are like 6 or 7 different phytosterols. They may be very different. Their ratios may be different. So pollens are very different. Don't think each pollen is made equal and everything is similar. So I'll talk about sterols in a minute. And then before I forget, I just wanted to emphasize nutrition is, as I said in the beginning, it's important round the year, but when they're raising those winter bees, that's when it becomes more critical. And probably some of you heard winter bees. This is a very loose term we use, but in the scientific literature, we use the term diutinous. So diutinous means long-lived bees. So the bees that are probably raised in the valley here between, I would say, we did some work. I'm not showing that work today, but we did some winter bee work as well to see what's the time period when they're raising those babies that can survive until March. So I would say anywhere between late August until October 25th is the time when they're raising those winter bees. And those are the ones that survive until spring comes. And most of them die by then. And your queen has already started laying eggs probably in January or February. So that population goes later. But those old bees are still there until February for sure. So those are bees that are really critical, right? If they have been, their immune system is compromised because of poor nutrition, then your diutinous bees will all die probably by December, and then there will not be much left in your hive. So that's what I'm saying here. The nutrition is critical. The protein is critical throughout the year, but then that's more important when they're raising those winter bees, because you want those nurse bees to have enough hypopharyngeal gland size so that they can produce enough food for that larva, and the larva is raised on a good diet, a proper diet, adequate protein levels, so that they can have a better immune system, and when they become adults, they will survive those 5 or 6 months of winter, depending on where you live. So there are lipids. These are again micronutrients. I'll focus on this steroids or sterols today, but you just wanted to say lipids are again a very broad classification. There are 4 different types within lipids. So sterols are really important. You need them in very small amounts, but they're really critical. So I gave you some functions of sterols here in insects, not just our bee. All insects need sterols, and we need sterols as well, right? If we are on Zoom, so I'm not gonna ask you questions and see how many of you are raising hands. And so sterol. Think about sterols for you, right? You also need sterols. Cholesterol is your sterol, right? So cholesterol, too much is probably bad because it might clog your arteries, but without cholesterol, all those important functions of cell membrane, those things will not happen. So you do need cholesterol, and that's a very important part of your composition. So insects also need sterols. It's a component of cell membrane. And the most important part is these molting hormones. So probably some of you still remember from your high school biology or whenever you took one, insects have to molt. They have to go into different stages, right? A 1-day-old larva has to molt to get to the second stage or day 2. From day 2 to day 3, again, they have to molt. So there are some hormones or involved there which are called ecdysone and juvenile hormones. And I will show you an animation in the next slide to give you a better sense on what these hormones are. So those hormones are really critical for insect growth. If suppose those hormones aren't there, then your larva will be just that 1-day-old larva will stay as a 1-day-old larva and it will desiccate, and then bees will either cannibalize it or remove and throw it outside the hive, right? So they're very critical for insect development. So all insects, including honeybees, those sterols are coming through their diet. For us, our liver makes your sterol, cholesterol, but unfortunately insects have to get it from their diet. And that's basically from the pollen that they are eating. So pollen is the source of sterols, and that's what those sterols are precursors of these molting hormones. That means without sterols, you don't have molting hormones. And there is this mouthful named 24-methylene cholesterol. That's a most important or a primary sterol for honeybees. And it comes through pollen again, as I said. Okay, so here you see I have an image where I'm showing you a larva and egg, right? So these are different stages that you see here. Are you seeing my cursor, Ian? Okay, good.
Yeah, we are.
Perfect, yeah. Okay, so what you're seeing here is these are all lying in that pool of brood food, right? So they're growing from one stage to the other. So from here to here, here to here, you need those molting hormones. And these are the 2, ecdysone and juvenile hormone. So there is an interplay of these 2 different hormones. If ecdysone goes up, then you are moving to the next stage. If juvenile hormone goes up, then you stay in that stage. That means juvenile means young, right? So those juvenile hormones will keep you at that stage. And when the time comes, the ecdysone goes up and juvenile hormone goes down, then you switch your test. You become a 2-day-old larva. Then you become a 3-day. So there is this beautiful interplay. I'm making it very simple here, but there are some other hormones involved as well, but these are the 2 main ones here. So if you didn't understand anything that I said, then just watch this animation. So that's your pollen, right? So I'm showing sterol as a pink dot here. So here come your bees. They will eat that pollen. So that sterol will get, sterol doesn't look pink. This is just for the animation purposes. So when the bee or the nurse bees eat it, it endogenizes. That means it gets into their bodies, right? You see those pink dots in the body now. And now she comes back here and feeds the larva, right? So larva also gets those because of the food that the bee has provided, the hypopharyngeal gland and mandibular. So here is an example of protein. So proteins usually don't have, right? This is only pure pollen, pure protein. So there is no sterol in there. So you don't see any of those pink dots. So the bee eats it and then she doesn't internalize much of that, right? Because there is no sterol in there. So she can't feed that sterol back to the larva. So this larva here doesn't have any pink dots. So there is no sterol. So you see it just desiccates, and then bees will either cannibalize or remove whatever they want to do. So that's why these molting hormones are really critical. And if you don't have the pollen, your colony will not grow because all the sterols are there in that pollen. Okay, so I'll switch gears and go over a study that we published recently. I mean, if you can use, it's an open access, so you can read if you really want more details here. But I'll give you an overview of the study in just 2 minutes. So we know that those studies in 90s and late 80s, they did at least confirm that this 24 methylene cholesterol is important for bees, but then we don't know what amount, right? We can put like a ton of it, or is it small amounts that you need for a colony growth? So that's what the whole basic idea was. What's the percentage we need in our diet? So this was a study done in the lab. So you can see all those cages there with bees, and we give them sugar solution in those jars and along with water as well. And then you can't see, but there is a small petri dish here that has the diet. So these are the diets. So the controls don't get any of this 24 methylene cholesterol. So here 24 MC stands for 24 methylene cholesterol. So there is another treatment S1, which has a 0.1%. This is 0.1% means 0.1% of the total diet weight, right? So 0.25% of the weight, 0.5, 0.75, and 1. So you may say, why did you choose these numbers? Because we have prior insect knowledge where most of the insects need somewhere around, it's always below this or above this. So we wanted to see what's our range for our honeybees. There is a story about this 24 methylene cholesterol. It's very expensive. I mean, you can get in pollen, but for these studies, you have to do controlled studies, right? You can use some bunch of pollen for the study, but then how do you control other things in the pollen, right? It's not just 24 methylene cholesterol. Pollens have other lipids, there are protein. So to have a more controlled experiment, you need to have an artificial 24 methylene cholesterol that you have to put in your diet. So the diet should be everything same, except for that cholesterol that you're giving in different concentrations. So that was a challenge. We looked to who is selling this 24 methylene cholesterol. We looked into Sigma, and there are so many other suppliers. None of them sell it. You can try Googling now. You will not find this anywhere. So this 24 methylene cholesterol, we found a Canadian company that was willing to custom synthesize for us. And it's pretty expensive because it goes through a lot of steps. If you have seen some chemists making compounds. So they took about 3 months to give us I think we bought like 5 grams probably, and for this study, very small amount. And it cost us about $80,000 just for that 24 methylene cholesterol. So my postdoc, who I said, Priya, maybe some of you probably heard her speak to your group before. She got an assistant professor position now in Mississippi. So when she was a postdoc with me last 4 years, when we were doing this study, when she held the vial, her hands would shake because if it drops to the ground, then you're wasting your $80,000, right? So it's a pretty expensive chemical. So don't try to use it. I'll tell you what to do in the last few slides, how to get 24 methylene cholesterol, but don't try to buy it from somewhere. It's very expensive. So here, this graph just shows you diet consumption. So you can see all this. Again, I'll make it simple for you to understand. You don't have to worry about all the graphs that I'm showing you here. So these are the higher concentration. So this is week 1. So I showed you all those different treatments, controls, then S1, S2, S3, S4, right? So you see, at least there is, so same letters means they're not different from each other. So these are different from the controls and lower concentration. So it looks like the bees are able to perceive the sterols that are present in that artificial diet that we are giving them. And they are eating those higher concentration diets more. That's a whole story in this graph here. Let's go to the next one. This is a mean head protein. So we take the entire head and look for protein content in both the hypopharyngeal glands and the mandibular glands. So you can see the higher diet, which was 1% sterol diet. You see it was significantly different from all these. So it looks like it is impacting their protein levels in the head as well if the sterols are high. And then we also looked at the abdominal lipids, and we didn't see a huge difference between the higher concentrations, but they were all higher than control. That means no sterol and lower concentrations of sterol. So survival, I know this is not an easy one to look at here. So you just have to believe me, whatever I say here. So here you see that orange one here, that's the S3. That's a 0.5% diet. So this is 1%, right? S5. So looks like 0.5% looks like an ideal or optimal concentration of sterol in the diet. So that's what the entire story of this study was. We think 0.5%, you don't need a 1% diet. 0.5% does the trick. You see higher survival and other things that we looked at. Okay, that's what the take home message here. Bees consumed larger quantities of diets that had higher 24 methylene cholesterol, and higher survival was observed in 0.5% concentration of 24 MC. Okay, so that's the study we did, but then there are some other labs that have been looking at protein and lipid ratio, right? It's not just simple like, okay, is 20% protein good for our bees? That's it, right? That's not the case. It's very complex. Nutrition is not that easy to really decipher and come up with some balanced diet that your bees like. That for humans, people are selling a lot of diets, right? For bees, there is nothing like a perfect diet at this point, but that's the goal. With all these type of studies that I showed you, or other people that are doing more nutritional studies, is to get to a point in the next 5 or 10 years that we have some optimal diet, even though it may not be a perfect diet, but at least we'll have most of the things that they need figured out. And then we can say this is something closer to their natural diet of pollen. So these studies have shown, again, this is not our studies. I'm just going to show you quickly. You can read these papers as well. They're probably open access. So here it says broodless nurse age worker honeybees consume foods to achieve a ratio between 1:2 and 1:3 for essential amino acids to lipids. That's what they're talking about. So these amino acids and lipids, they need in a certain ratio. It's not just that lipid is there, you're okay. They have to be in a proper ratio as well. So there is one more. I know probably some of you take fish oil, right? Because they have omega-3 fatty acids. So omega-3s are important for your immune system and bees' immune system as well. So there are a couple of studies done in Israel where they have shown this omega-6:3 ratio. It has to be close to a 1. So then I think the bees do well. So again, so we'll be looking at this. We haven't looked at omega-3 and 6s yet, but it's in the plan. In the next few years, we will be looking at the requirements of these as well. But there is some work already done from Israel. Again, this is a paper you can read, Frontiers in Psychology, I guess. So these are important as well. I'm just telling you there are so many unknowns at this point, and these are all micronutrients, but they're very important for insect immune system, especially in our case, our honeybees. Okay, if you have your Slido thing, if some of you have downloaded, and that's the number if you want to, and then looks like it should be open for now, right? Is it opening? I don't know if Ian or Jenna, you have opened. Okay, looks like I'm getting some results. So I'm asking you a question. Do you feed protein supplements to your colonies? Okay, 50-50 at this point. I'll wait another 30 seconds if you want to. Oh, okay. So there are more people not feeding, huh? Okay, just keep changing. Okay. Okay, I think we got about 60% say no, and some say 40% say yes. Okay, that's good enough information. So I know there is no need to feed if you think you live in an area where you get a lot of pollen. So it depends. Protein is not fed throughout the year. Usually even commercial beekeepers, they will feed when there is a need, right? Especially if there is a protein dearth. That means there's not much pollen coming in like late August, September as well, because the bees are still raising their brood, right? They are still raising those babies, and you need them to be in a better immune system. So you want them to get some pollen. So of course, protein supplements are not anywhere close to your pollen. They're missing all those micronutrients that I talked about, but protein is the major one they will supply. So most of these diets, if you buy anything from Mann Lake or Bee Pros or whatever global patties, there's so many in the market. So they all have a decent amount of protein. I would say anywhere between 15 to 25% or even more, some of them. But you don't need too much, like 40% is an overkill. You don't need 40% good protein. Anywhere between 20-25 probably will do the trick as well. So you see here from a commercial beekeeper, I took this image. You can see they feed about 2-3 pounds at a time, and they probably, some years I have seen them even feeding 8 to 10 pounds of protein supplements to their bees. But you don't have to. As a backyard beekeeper, probably you live in a place where there is some constant source coming in. And these beekeepers, they go for pollination, right? Some crops are very notorious. They like blueberry especially, they don't get much pollen when they are in blueberries. So they have to feed sometimes, otherwise the colonies will decline. And there will be another example would be carrot seed. If you have ever gone to Central Oregon, they grow about 5,000 acres of hybrid carrot seed in Madras area. And that crop is also not so nutritious for bees. It doesn't produce enough pollen. So beekeepers really struggle there when the hives are there, because the colonies are declining in size because of lack of pollen. So in those times, they do feed protein supplements, at least to stabilize the colony growth. So there is value to feeding protein supplements, but again, as I said, they don't have all those micronutrients that I talked about in the last few slides. So yeah, as a beekeeper, it's your judgment. If you think your colonies are not struggling, you still have enough pollen in your hives, then probably there is no need to feed protein. But if you live in locations where there is a pollen dearth, and if you see a month or two there is no pollen coming in, especially when they're raising those winter bees, then probably that's a good idea to feed some protein supplement. And some of them, they will sell 5-10% pollen mixed in. I think I'll talk about that in the next few slides. So these are some challenges that our bees face when you have, so these are more probably applicable to commercial beekeepers, but it could be you as well. If you live in a place where probably there is not much coming, or we are more close to some intense agriculture. So lack of pollen abundance is one thing. That means enough pollen isn't coming. Lack of pollen diversity. Diversity is also important, right? As I said, all pollens are not made equal. So if suppose there is an amino acid absent in pollen A, it might be present in pollen B. That's why when you see bees have evolved, this is a very beautiful evolutionary process. You sit next to a hive, bees are not just bringing one color, right? Sometimes you'll see 4 or 5 different colors of pollen coming in. And unfortunately, bees are very good at figuring out concentration of sugar. So if I put them 5 different petri dishes with 5%, 10%, 20%, 50% sugar, you will see eventually they may be all random in the beginning, but after 20-30 minutes, you'll see more bees on the 50%, the highest sucrose solution, because their antennal receptors, they have the ability to detect concentration of sugar, but unfortunately they don't know protein quantities. So if they have never encountered a pollen in their lifetime, the species that you're going to give, you give them 5 different ones. And because you as a beekeeper or a scientist, you have done some analysis, you know this pollen 5 has highest protein, but bees don't know that. They will randomly collect pollen. So that's the thing I'm talking about. This pollen diversity is also important, if not too critical, but it is important just because they can balance the nutrients that are absent in other pollens. And lack of pollen quality, like I gave you this hybrid carrot seed example earlier. So lack of pollen quality means basically what is missing, the protein levels, right? It might be 5% protein when compared to your tree fruits, like all your pear, cherries, they're about 20 to 25% protein, which is a decent amount for a plant species. So anything less than 10 is probably not a good pollen in terms of protein levels for bees. So those are 3 things you should be thinking about as well. So so Ellen, when she was doing a master's project with me, we published this work like a few years ago. So we looked at pollen diversity available to bees in several major cropping systems here in the West. So I'll quickly go into this because as a backyard beekeeper, probably you're not interested in this because you don't go for pollination. But of course, it relates to you depending on where you live, right? So I'm just giving you a very broad overview of the study. So you see here on the left, you have the crops: almond, blueberry, carrot, cherry, and meadowfoam. So you can see the different plant taxa that were there. So we put pollen traps on these hundreds of hives that they were in different cropping systems. And then we analyze the pollen from the pollen trap. So it's pretty tedious work. And yeah, so we had a collaborator at Texas A&M who was doing the pollen IDs for us. So here you can see the almond. So when almonds bloom, nothing else is in bloom. Probably some wild mustard in California and maybe some dandelions. So that's why you see there is not much diversity. It's all mostly almond pollen coming in. When bees are in blueberries, there are 13 different species, including like viburnum, there is Scotch broom, several noxious weeds as well. So that tells you that the diversity is high, but then you may think, oh, then blueberry bees should be doing well, right? Because the diversity is high here. But when you look at the quantity here, you see how much small quantity they're bringing in. So it is more important to have quantity as well, not just the diversity. So bees are struggling here. They can't get everything from blueberries. That's why they are visiting so many other species around those blueberry fields. So the diversity is high, but the quantity that they're bringing in altogether is very small. So that's what I'm trying to show you with the study is just one thing doesn't always make the trick. So you have to have enough pollen and then have enough diversity as well. Okay, so that's enough to give you a sense. I think I talked about all the basics. Now I'll switch into this habitat improvement, right? There is a lot of push in the last 10 years, I would say, to improve habitat for bees. And even Xerces Society has a list of plants that people can plant. And looks like there is a speaker from Xerces going to be speaking somewhere in your group, probably at the end of end of this year, probably. So you may get some insights from them as well on what they are doing to improve habitat for bees. So, and you probably, I don't know, maybe if this image is from Portland somewhere, I don't know. I stole somewhere if I have their name here. So this is very common, right? In big cities, wherever you go, pollinator habitat, that looks like there is some California poppy here and something else. And then we are also trying in many areas, like this is the urban, this is the ag setup, right? You have carrot seed crop here. So we convinced some farmers in the past to, this is bachelor's button that you're seeing here, to plant something like, this is just an example. I'm not saying bachelor's button is the best thing for the bees here, but something that they can still easily plant and this doesn't become a weed for them. So convincing farmers is really hard. If try telling farmers to plant something for bees, because they're already paying money to pollinate the crop, right? They're paying $100 per hive or something like that for hybrid carrot seed. Why would they want to spend a bunch of money if on irrigation and then weed management and all extra work here to plant something for bees? But now there is a paradigm shift in thinking, right? There is a lot of awareness about pesticide exposure and the importance of bee health. So I think it's a relatively easier time now to convince farmers than it was 20 years ago. So that's an effort which we are doing in some crops here in Oregon to see if we can improve some habitat for bees so that bees have some natural pollen coming in. And the other question you can think as a beekeeper, and that's what the farmer will ask you always is, right? So they'll say, you are planting or asking me to plant all these things. Would the bees get distracted to this? And because carrot is not a great crop, then do I not lose pollination, right? That's a good question. So we have tried to give some biology explanation for that is, I mean, there will always be a distraction, right? If you put like 50 hives here, they are not going to be here. There will be probably 30–40% of them are going somewhere else too. You don't have much control on what you can do with bees, right? So that's what we have been doing with, so I think that I have that slide somewhere else, but let me talk about what we have been doing with something related project here. So this is a USDA funding we got last year. It's a 3-year project where we want to develop a pollen nutritional composition database. So I saw Glenn somewhere in the audience as well, and probably I have heard him. He does a great talk about all the species that are in bloom. I don't know if he does that still for your group or not. Bloom updates or whatever he calls them, right? So we have all these beautiful plants we know, right? They bloom at certain times and our bees have access to them. But then when you look at NRCS or Xerces website, they will have a bunch of list of plants, right? And probably as beekeepers, you get this question quite often is, give me top 10 plants that I can plant in my backyard that my bees will really do well because of those, or something like those. And I get that question all the time as well. But it's unfortunately, I know we can give some suggestions. I do still give some, but wouldn't it be great to give them based on more scientific evidence on what is there in the pollen, what is there in the nectar, right? Or what volumes of nectar is produced from specific plants. So that's what we are trying to do with this grant is look for at least 100 or 150 different species of plants that are attractive for bees to quantify or at least look at the nutritional composition of those pollens. So let me give you an example here. Not a great analogy, but suppose you give me French fries and salad, right? I'm gonna probably try French fries first, right? Because it's attractive. So does that mean that, so let's think about your bees. If they have something like French fries for them in their world, some inferior pollen or inferior nectar, whatever you want to say. So that doesn't mean that it's just because attractive doesn't always equate to a good diet, right? That's what I'm trying to make a case here is, does attractiveness always translate or equate to best nutrition? Probably in sometimes it may, but not always. So that's the whole idea of this is we want, and most of those recommendations now on those plant lists we have, it's based on attractiveness. I'm not criticizing that. I'm saying that attractiveness is the way to go for now. but wouldn't it be great to have additional information apart from attractiveness? So that's what the next slide I'm going to show. So this is an NRCS one, USDA. See all these beautiful lists, and they have these PDFs where you can go and find out more about those plants, when you can plant, what type of irrigation is needed, how they do in different regions, and all that. It's great information. And same like same, you can look at Xerces Society. They have a bunch of plant lists for different regions. And so what we are envisioning is to have something here, like another column where I say pollen nutritional composition. We will put crude proteins, sterols, lipids, omega-3 fatty acids, whatever is possible. So I'll take pollen from, suppose, let's see it here, it says big leaf lupin, right? Lupin. And then we can say, okay, so this one is great. You have all the information. Now I'm giving you more additional information on pollen nutrition as well. So that makes you make more better judgments on what you want to plant in your backyard. We are not saying, oh, don't do this, or maybe within this you like 5 of them, then you have additional information here to make a better judgment in a more scientific way, right? So that's the whole idea. It's a very ambitious project. We are again, I'm going to show you a slide and ask your help as well. Probably you heard me or Priya or someone from the lab say that before as well. We want to collect enough pollen from different species. So we're looking for a gram. Gram is too much, sounds small, but try to collect. It might be thousands of flowers depending on how the species is and what type of, it's an open flower versus something like your blueberries, which are more bell-shaped, hanging. So it's very different. So it's ambitious project. We are trying to get at least 100 different species this year and next year as well. So that's the whole idea. So watch this. So this is by collecting from bees. If you can't hand collect, then this is the way. Okay, so yeah, if you're interested, email me. That's my email, or you can contact Ian or Jana. They can email or forward your email to me. So that's how we collect from bees. But then you have to have a significant acreage, right? If you have only 10 plants, probably it will not work. We are looking for 50 foragers. So that gives you 100 pallets, right? So each bee have 2 pallets on the back of their legs, on the back legs. So that would amount to about a gram. So, but then we have designed a vacuum, like one of the graduate students, Max, he has designed a vacuum with several meshes. It's a handheld vacuum. And if it's an open flower, easy to suck pollen, then we have collected enough pollen from that too. So yeah, if you want some ideas, I can give you, but if you think you can rely on bees like this to collect them and put them in the freezer if you don't want to kill them. But if you don't mind killing 50 bees, then you can stick them there until they die. And then you can remove those pellets from their legs and put in a centrifuge tube and put in your freezer before, and then yeah, it can be outside because protein degrades if you put in that room temperature. So they have to be in -20 until you ship them back to us. So yeah, if you're interested in helping with any 1 species, 2 species, whatever in the next 2 years, please email me and I will send you more protocols and more videos to watch on how to do this, or I can do a one-on-one call and give you more details as well. But we are using both manual collection with the vacuum or sometimes some like sunflower is the easy one, right? Probably we don't need sunflower now, but you can bring heads home and put in a flower vase in water overnight on a white sheet, and you'll see a bunch of pollen dropping overnight. So those are easy ones to collect, but there are some painful ones. We're trying to do some camas this afternoon, and we couldn't collect enough. We got like 0.1 gram after a couple of students working for 3 hours or so. So some are painful ones, but unfortunately we need a gram to do all those different things that I mentioned, from protein to lipids to sterols to omega-3 fatty acids and others. So yeah, we'll see. I think that might be a really good information for people, not just beekeepers, but all the land managers or the conservation groups that really are promoting habitat for bees in the future. We think that's a more scientific way to get hold of some top 20, 30 plants to plant. Okay, so this is a slide where I wanted to convince farmers, giving some bee biology thing. So that's what we usually say is supplemental forage. Like I showed you this bachelor's button planted next to carrot seed crop. So it not only improves bee health for beekeepers, right, but it also increases the crop yield as well. And that's what we have to convince them is if that colony is losing size, that means the queen has stopped laying eggs, then it's no win-win for both beekeepers and the farmer as well, because the pheromone is the most important one. The larvae are releasing a pheromone telling the bees to get more food. Could be pollen, could be nectar. And if the bees are not getting that stimulation, they will just sit idle or they just will not do anything, right? So that's how the crop yield may be impacted as well. So these are some things we have been trying to explain beekeepers and farmers to see that it's a win-win for both if we have supplemental forage for bees. I'm not asking them to plant, if there is 10 acres of carrot, that farmer doesn't have to plant 5 acres of supplemental forage. We're looking for even 1/4 of an acre. That would be more than enough to get at least the pollen that they need. So that the colonies don't decline, because the queen stops laying eggs if there is no brood in there. So the pheromone is a cue that's both for queen and the workers too, that they need to bring more forage. So this is a slide that probably some of you want to try convincing a farmer or someone to plant habitat. So that's what here you say optimal nutrition, right? So you have good nutrition. That's why you get all this nice looking larvae. So they are all releasing a pheromone. That's how bees know. Bees don't check on each cell and say, oh, we have 2,000 larvae, so we need to get this many grams of pollen. That's not how it's dark hive. They can't see literally anything there. They are perceiving all this information from pheromones. Pheromone cues, I mean, those new beekeepers in the audience, pheromone communication is very critical for bees. They do a lot of things based on just chemical communication and those pheromones. And there are so many pheromones, from queen pheromones to larval pheromones. And so this is a brood pheromone that the larvae release and tell the bees that you need to bring more pollen for us. So if you remove a frame of brood, you will see the number of pollen forages will reduce, because they know there is less babies to feed now. So people have done all those type of studies. And so for my PhD, when I was doing at Texas A&M, we did a larval pheromone, because we know the compounds in there. These are all fatty acid esters. So we know what exactly. So we have this 10 blend of fatty acid esters we put on a glass plate and then put in a hive, and within hours you can stimulate foraging. So you're fooling the bees with extra pheromone in there, saying that we have more larvae, so you need to get more food for them, which probably is not an ideal thing to do, because you're confusing bees and creating a little more colony health problem there. But for pollination, sometimes you can use those pheromones. That's an apiculture use for enhancing pollination for short term, not long term. So again, coming back to this slide, you see there's more larvae. That means there will be more pheromone in there, and that's what you see here. It stimulates more foragers to get food. So you'll see a lot of foragers coming back with pollen now. So that increases their pollination as well, right? So they're promoting pollination but if they don't have, if the farmer doesn't plant anything, so they will be tired of these carrots after some time. There's not much pollen coming in. Then you will not have these larvae much. We may have small amounts, but the pheromone is not enough for them to forage aggressively to get more pollen. So it's a win-win for both. That's how we have been convincing farmers, is if you explain them a little more biology, because when they rent bees, they have zero knowledge of bees. They don't even open those boxes to see if there are any bees in there. So it's just something more educational for farmers that are not much aware of the bee biology, to explain that these babies are really critical. They are the ones telling these adults to bring more food. And that's how the pollination increases as well. Okay, so this is the most important slide, I would say, for new beekeepers here. So make sure your bees have access to adequate pollen throughout the brood rearing season. And there are companies that will sell pollen mixed with protein. So that means it's not pure protein supplement. There will be some 5 to 10% natural pollen, and that has your sterols in there, right? So you need some natural pollen mixed in. You can sustain your bees for a month or so with straight protein supplement, but then they will miss on all those vitamins and minerals and those sterols, all the micronutrients that I talked about. So you need those. That's why you need some pollen for sure in your hives. And as I said before, it's important that they have good diet when they're raising those winter bees, especially fall and late fall. And I get this question from beekeepers, so I put a slide here to show if you can trap your own pollen, right? Some time of the year, there is a lot of pollen coming in and bees may not need entirely, so you can trap some pollen. So they sell these traps. So this is a front porch trap, a yellow one. I don't like them much because they are plastic and sometimes they may damage the wings of your bees, but you're not putting it for too long. So probably you trap for a day and then you remove and then you put it back again after a week. So you have to look for very strong colonies. Never put these traps on weak colonies. Then you will kill those because they're limited in pollen, right? So that's what I say here. Select strong colonies, and then preferably the ones that are getting morning sun. If you have some 10 colonies and some of them may get morning sun initially, right? So you can see always a colony that is facing the sun becomes more active in the morning sooner than the one that is facing the other direction because of the sun, right? So they use these front porch traps. Before you decide a colony, there might be some holes, right? If you have an auger hole here like this one or some cracks on hives, you may have to seal everything because bees are pretty good. If you put a trap here, they will try to go from those holes and then it defeats your purpose of collecting pollen. So seal all the entrances. So install the trap and wait at least a day to engage the trap. When I say engage, you see this screen here, right? So this actually, it folds up. So you can fold it. So if you push this in and put a tape, it will just stay there. So your bees can directly go without going through those holes. So you have to orient them first. If you start like in the middle of the day, go and put this thing, you're confusing them totally. And then some of the bees may fly into other nearby hives because they know something is wrong here. They can't get in. So always try to put this maybe in the evening when all bees are in, or maybe early in the morning before they start. And never close it, just keep it open for a day. Then they will all orient, right? They will know my weird beekeeper has put some yellow stuff here and they'll start going in. But once they get trained, then you can drop this thing on next day. They will come through this in the morning and they will orient. And that's a better way to trap pollen. And you don't need tons of pollen. I'm looking at maybe 3-4 tablespoons from each of the hives that you have strong ones. And pollen can be stored in the freezer without losing any quality for about a year. So store that pollen in a Ziploc, put it in -20, and you can use it next year as well. When you're feeding your protein supplement, just smush it and mix it with your protein supplement. So that could do the trick. So there are, again, I'm just very quickly going through this, but yeah, so there are tips here, but there is a manuscript we published last year. This is also an open online one. You can just Google my last name or Ellen Toppert-Zoffer and say collection and identification of pollen. So you'll find there are a lot of tips in here if you want to trap your own pollen. And we also have a publication. We did a review for veterinarians because veterinarians are interested in bees now because beekeepers like you, if you need a treatment for EFB, I don't recommend using too many antibiotics, but sometimes you may have to if the disease is a big problem in your bees, in your larva. So veterinarians wanted a publication on bee nutrition as well. So we published this last year, I believe, but it might be useful for you to read as well. So it just very briefly goes over all different topics of bee nutrition. And it's an open access, so you don't have to subscribe or anything for this. Okay, so that's about nutrition. You can take questions now, Ian, or you want me to take 5 minutes to finish this one too?
Yeah, let's go through the entire thing, then we'll hold for questions.
Perfect. Yeah. So I think I spent like 45 minutes. I'll spend another 5 minutes and I'll stop after that. So So European foul brood, again, I don't know how many of you have seen. So I think I have a question here. Go back to that Slido thing again. And answer this question. Have you seen EFB in your hives past year or this year? And for new beekeepers, you don't have to if you don't know about it. It's called a European foul brood disease. It's a larval disease. It doesn't impact adult bees. It's only a larval disease. And some years we see it pretty bad, but some years we don't see that much. So, but last few years I have seen commercial beekeepers are struggling with this, especially in April, May, and then even it lingers until June as well, some cases. So okay, looks like we are in good shape. You haven't seen, or maybe you don't know that's EFB. You don't know. Okay. So that's good if you haven't seen. But yeah, if you see, let us know. We are doing a study to figure out, is this a new strain coming or is it something of a real problem in Oregon and many other states where EFB has been increasing in the last 5 or 6 years. Okay, so potential factors. So why we think EFB was supposed to be always a minor disease. AFB is a notorious one. For new beekeepers, American foul brood, yeah, if you have that one, yeah, of course, you have to be really, really careful. I mean, the best recommendation is to burn your hives. You don't want those spores around because American foul brood is a notorious disease. People have shown those spores to be viable for 60, 70 years as well. So that's why the recommendation is to burn hives if you're a backyard beekeeper. But commercial beekeepers, I know the disease is everywhere, all continents now and every state here in the United States. So those who have 10, 15,000 hives, they don't burn the hives. They usually try to use antibiotics. So unfortunately, they are on this vicious cycle of antibiotics, of this treadmill of antibiotics. So when they stop using antibiotics, the disease shows up again. So that's a different story with commercial beekeepers. I'm not saying all beekeepers have it, but there might be some that may be masking the disease by using antibiotics. But I understand their point as well. Okay, are you able to destroy 10,000 hives? So it's an economics issue as well. So these are some stressors that people have been hypothesizing. Maybe it's a nutritional stress that causes the disease. Is it a new variant? Because no one has looked at these variants. Like we talked about COVID, right? How many variants have come now? So same thing. Bacteria, viruses, they evolve to the next stage with mutations and all that stuff that you know. So there could be a new variant. We don't know because no one has done in the last 10, 15 years a proper study to look at has EFB varied or the variants have come in the last 10 years. So that's one question. When bees are in blueberries, mostly that's when beekeepers complain. And some of these crops use fungicides. So there is a hypothesis, are fungicides a problem? Are they creating a stress on larvae and creating a bigger mess with this disease? There is another very minor hypothesis, I would say, not so credible. It says acidic pH of blueberry pollen. So there was a researcher who did some work in 80s, I believe, or 70s. There is a thesis. It's not even a published work. They hypothesized that blueberry pollen is a little acidic. Maybe that's why when bees are getting blueberry pollen, but then with the study that I showed you before, with the Ellen study, the graduate student I had, we didn't find too much blueberry pollen coming in to the hives because bees don't go for getting pollen. They're only getting nectar from blueberries. They're not getting much pollen. So I don't know if that hypothesis even makes any sense here. And then brood chilling. April, May are usually colder months. So there could be a point that there is less ability for bees because bees are still growing, right? If you haven't gone to almonds, your bees are still smaller in size. I mean, the colonies. So probably they're not able to maintain temperatures in the larval brood area. So that might also cause a stress because of cold temperatures. So those are the ones. So this is a study. We got some funding from Blueberry Commission here to look into that because their goal is because beekeepers are blaming blueberries for this. So they want to know, is it a problem when bees are in blueberries? So we are looking at all these things. So we want to look at the prevalence and intensity of European foul brood disease. We want to look at, if we find, then how do we mitigate it? Is it by just feeding protein supplements or probiotics? So we're looking at all those different angles. And then we also collect samples when we find these EFB to see, So I have a collaborator here who does whole genome sequencing. So we have a sequencing facility that can look at all these variants. Are we finding different variants in different locations? So that's the whole thing we want to look at. So these are the ones that probably to look at is nutritional state of the larva, right? That's important because the colony protein availability, if there is less pollen coming in, that creates a nutritional stress on larva, and that could lead to a disease. That's what I mentioned before. And then you should also think about ratio of larva to nurse bees. I don't know if you heard of this term before. So you need a proper ratio. There should be enough nurse bees to feed the larva. If suppose you have 2,000 larvae in the hive, I'm just making up some numbers. You should have about 4,000 nurse bees feeding those larvae, right? But in spring, what happens when beekeepers come from almonds, they are making splits. You all probably know what splits are, the new beekeepers. So they're dividing those colonies, right? The huge colonies, they're making splits and creating new colonies here. So in that process, they may be skewing the population. They are not very meticulously, oh, I have 2 frames of nurse bees here. It's hard to say that, right? So they're stealing frames here and there and shaking some bees. And so that process may skew this ratio of larva to nurse bees. So suppose if you're skewing that, that means there is more larva, less nurse bees. That's a problem because now the larvae are not getting the diet they want, right? So that's how there is a nutritional stress on bees and that on larvae, and that could create a more problem with EFB because EFB is going to be there. It's an inoculum is there, but the larvae don't show up the disease unless there is something wrong with their immune system because of all these stressors that I talked about. So this is a study like you see the students there in blueberries. We have been sampling. We are continuing this year as well. These images are from last year. See that's blueberries again in the background. So then we also compare them with hives in other crops like clover that you're seeing in the bottom one here. That's a turnip on the bottom. That's blueberries again on the top. So we have been taking samples of pollen. So this is a trap. You can see how, so if you are using a front porch, that's how the pollen drops into the tray and that's what you store. So we are looking for any fungicides or other pesticides in the pollen when they are in these cropping systems. So this is how you fit those pollen traps. So you see these are commercial hives. So one of the beekeepers that we are working with. So you see we put 2 traps on the same because we learned this the hard way. So if you don't put one, so suppose there is only 1 trap on this, all these bees are, so these bees are confused. Half of them will go into this hive because there is no trap. So when you have hives on a commercial setup like this on pallets, it's always a good idea to trap both hives at the same time, that you're equally confusing both. So then these bees will not go here or here. So I know you're all backyard beekeepers, you will not encounter this, but if you happen to be having pallets and you're trapping, then of course you have to put 2 traps at the same time. So we do a lot of colony evaluations in those hives as well. So these are the swabs that we take with the larval swabs to look for those different variants by sequencing. I think I'll stop here and take questions. I think those are the 2 topics I wanted to finish today.
Awesome. That was super interesting. Thank you, Ramesh.
Yeah, no problem.
I have some questions, but I'll let anybody else, you can put them in the chat if you'd like, or unmute yourself and ask away, please.
Most of them sleeping?
They're probably outside playing with their bees since this is a nice day out today.
That's fine.
I'll ask a question. Ramesh, you thanks so much for your talk tonight. It's always so interesting to hear the science that kind of backs up what we do and why we do it. I know I'm guessing you probably won't recommend a particular pollen, like if we had to do a commercial pollen patty, protein patty with pollen, do you have a range of recommendations or several brands that you
that's a great question. I know as a researcher I'm not supposed to advocate for any of these companies, but I think I can indirectly tell you what we use for our studies and other things maybe. So I mean most of them have that. As I said, all brands have decent amount of protein, the total crude protein, and I don't think anyone is trying to add all those sterols and other things I was talking about. So I'm just suggesting use someone that uses a 5 or 10% pollen mix. So the only company I am aware of that has that 5 to 10% mix is a Global Patties. I don't know if you heard of, I believe it's a Canadian company, but I think they have a brand somewhere in the United States as well. Global Patties. Have you heard of it?
I think so, but I'll have to Google them. Thanks.
Yeah.
I really appreciate that.
Yeah, many commercial beekeepers use as well here.
Yeah.
That's how I came to know about it, because a couple of commercial beekeepers like 10 years ago, they were using, I think they make it, even the texture is, that's important as well, right? Some of them, they just dry up after 2 days and bees struggle to eat those. So looks like they have this consistency as well, which is probably stays like that for a week or so until they really consume it. So I think that's another important aspect to look at. If some of them, they dry up like rock, then they're not useful, bees will ignore them. So it looks like this brand, at least the ones that we have fed in our 2 bees, they look to be pretty decent even after a week. And they do sell with 5%, 10%, even 15% pollen mixed.
And so are you recommending, supplementing that most of the summer? Because you're talking about the brood rearing season, kind of.
Yeah, no,
pretty
we don't feed,
consistently?
we don't feed like our OSU hives. We feed only mostly probably late August.
Okay.
Because until then we have a decent supply of pollen coming in from different things.
Okay.
So for nectar and pollen, I think blackberries, your Himalayan blackberry, those new beekeepers here in the audience, Himalayan blackberry is a major source of nectar for bees here in the valley. There is so much Himalayan blackberry, it's a weed, but unfortunately or fortunately, that's a major source of nectar in either June, depends, each year is very different, right? Some years it's done by June, but some years it just keeps going depending on the temperatures. I have seen even them blooming until July 15th or 20th. So they get some pollen also from it. So it depends on where you live, but in our area, I would say we don't feed any protein supplement until like August 15th or so. But then, yeah, depends on, again, if they're, your bees will tell you the story as well. If you live in an area where they are bringing in a lot of natural pollen, you will see that pound or 2 pounds that you put in there, they will take a long time to eat, and then you'll see them ignore it. So that means that they are getting their own natural pollen. But if it's all gone in 2-3 days, that means they really are looking for protein.
Ramesh, there are some questions coming in on the chat. Do you want to look at those, or do you want me to read them out?
Yeah, you can start reading, but I can look as well. Yeah.
Okay. The first one says, might the bees avoid collecting blueberry pollen because it is acidic?
So what I know based on what we have done in blueberries is, the flower structure is a pain. You see honeybees, they struggle to get in. The bumblebees have a longer tongue. They do a lot better job of pollinating blueberries. And that's why, and you probably heard of that buzz pollination, right? Bumblebees have huge thorax. They can buzz, and that's what blueberry pollen will not come out until you shake with either a toothbrush, like an electric toothbrush. It has to be really vibrated. So, but the bumblebees do that vibration, whereas honeybees, they just gently go and sneak in and try to get some nectar. So the pollen has to come to their legs, right? So they don't really try to do that. So I have talked to growers, blueberry growers. They say, oh, so you're saying blueberries are only giving, like, when you put a pollen trap on those hives, you get like 4% pollen from blueberries. So then the farmer thinks, oh, then we are not getting any pollination. No, what we did is we caught some nectar foragers. This was like 7 years ago. We did a study. So you catch those nectar foragers when they're getting into the flower, and then you put in a small centrifuge tube, bring back to the lab, and then you do a total body wash on them and then look for pollen grains. So you do find pollen grains on the nectar foragers. So nectar foragers are doing the predominant job of pollination in blueberries because it's a painful flower. Only the nectar foragers go and use their tongue and try to get some nectar. But the pollen foragers, they can't get in all the way to put out the pollen on their legs. So they avoid blueberry. And that's the reason we think, not just because it's acidic, or I still don't believe that hypothesis. This was one thesis that was done in 80s. So people have caught on that, but I don't think there is much evidence of blueberry pollen acidic, and that's why bees don't collect. It's mostly the flower structure is a painful one for them to get in and get the pollen.
Thank you. The next one says, is there a harm in keeping sugar and patties on hives when there is no clear shortage of pollen coming in?
Okay. So, I mean, there is no harm. Of course, bees know, right? They protein is attractive to them, but then if there is natural pollen coming in, that's what we have seen with our studies or with our experience, they will ignore your protein supplement if it's there. I don't think it does any harm. You're just wasting your money by buying the protein supplement and putting on that. But I don't see any negative consequences having a protein there. If there is a real shortage, they will start consuming the protein supplement. So again, I can't speak for everyone because you all live in different locations. Even a microclimate where you live in Portland or outside Portland, it depends on what is there in bloom at a certain point of time. So yeah, so just with your experience, you can try, just use maybe a half a pound and see at a certain time when you think they may need. And if they're ignoring it, that means they still have enough pollen, adequate pollen for their colony growth. But I don't see any harm in having it on top. They will just ignore and not eat it.
Does color of the pollen have any indication of protein or sterols or anything else like that in it?
Yeah, that's a good question. So with color, no, we haven't seen any correlation with color versus the color only helps to some degree to identify a pollen to some degree, but other than that, I haven't seen any correlation between color and the protein content. Looks like there are more questions coming.
Yeah.
Okay, it says if using pollen patties to supplement, is there a sterol that can be added to the bee skid? Okay. Don't buy the one I told you. That's very expensive. So that's the whole goal. We want to see if I get this question quite often. Are you looking for a source of sterol? I mean, we know pollen is a source, and almond is a great pollen. We have seen almond pollen has probably one of the highest sterols. I can't get the number straight at this point, but we see a lot of sterol in almond pollen as well. But then you have to feed quantity, right? We don't know exactly how to. So there is this, there is a lab that is looking, it's a USDA lab. Have you heard of spirulina, the algae? So spirulina is having a lot of 24-methylene cholesterol as well. But then we don't know if you just feed this algae to bees, do they even digest it? It's not their food, right? So there are so many consequences of those. So digestibility, assimilation, does it really transfer into 24-methylene cholesterol and get to their bodies? And so there are so many questions. So that's another angle that people are pursuing, including us, to see if there is a source that we can find, a cheaper source, because growing algae like the spirulina is very easy, right? It's not that complex. Someone can grow in a facility if they have the abilities to do that, then that can be used as a 24-methylene cholesterol sterol. But unfortunately, at this time, you have to go with pollen. That's what I was telling you, to trap your own pollen. So I'm looking at maybe like 2 tablespoons of pollen would do the trick for your colony, that single colony that you want to feed. You're not looking at, that's what I said, it's 0.5% of the diet, right? So just to give you a simple idea here, 2 tablespoons probably each time that you're feeding, maybe twice in a year, that should suffice their needs of sterols. But yeah, we don't have anything at this point to really suggest that you can buy and add sterol to your, except pollen. Yeah, pollen is your only choice at this time. But maybe in the future, you will find something. Will supplements, what is that, C cholesterol or sterols be available soon? Yeah, that's what I think I said. There is a scope, but yeah, we don't know. There are several other labs also trying to get some of this 24-methylene cholesterol figured out. So if we find some alternate source, maybe that might be the way to go. Have been growing meadow foam last few years. Yeah, meadow foam is also a good source of pollen for sure, but we haven't looked at the sterols and other things. But I really like meadow foam. I think it has a lot of promise. For those in the audience that probably have not seen meadow foam, it's not grown everywhere. If you are on highway on the I-5, maybe south of Eugene, Harrisburg, that area, if you see something very bright white in May, that's probably meadow foam. It's a very beautiful crop, and bees love it. If it doesn't rain, they collect a lot of pollen from meadow foam. And meadow foam is for oilseed. It's an oilseed crop. They use for cosmetics. There are a lot of things they do with the oil. It's a very regulated crop. They grow about 3,000 acres of meadow from here in the valley.
Cool. Any other questions?
Nope, another one just came in on the chat. When you're inspecting your hives, are there any signs that your bees are having malnutrition?
Yeah, I totally forgot about putting a slide there. Actually, I always have in my talk. I don't know why I missed that one. So if you watch those larvae, but that's an extreme event, right? It's not easy. The quick answer is it's not easy to figure out starvation. But then if you have looked very carefully at your larva when you open a brood frame, if you don't see too much brood food in there, they're literally, let me see if I can find it while I'm talking, if I can find that image. So if you see they are dried up, they should be always in a pool of brood food, right? It should be enough, like it should always be, the larva should be almost floating in that brood food, the white stuff that you see in the cells. But if you see that it's totally dried up, you barely see some and they are just trying to move around, because larva can move. You probably think, oh, larva can move. They will. That's how they adjust their angles. They're very small movement. That's why if you don't have food, they will try to go to the sides to see if there is any food around. So, and then if you are try to see if you want to prove that larva move, bring a frame of bee larva that are like 4 or 5 day old, like the chubby ones, and put near your sink. Within a couple of hours, they will all crawl down and fall into your sink, because they are barely, they know that there is something wrong. They're trying to find the food. Where is the food? The nurse bees are not feeding us. So as long as they're being fed, they will not move much in their positions there. But once you stop feeding them or you bring the frame into your kitchen, they will start crawling out. Very slowly they wiggle. I have a couple of videos, not for today, but maybe next time I speak, I'll show you some other videos too. But yeah, maybe I'll share that image. I can't find right now. I don't want to keep you here too long, but I'll just send that one PowerPoint slide to Ian and Jenna, and you can share with whoever was asking. Is it who was asking the question? Bruce, I guess.
I think it was Bruce.
Yeah. So yeah, there are ways, Bruce, you can look and see. That's a very drastic one. If they are really literally struggling, then you will see that food is not there in those cells. And that tells you that these larvae are starving and maybe there is something wrong with pollen. But otherwise, you have to send a sample to us. We do something called a Trehalose assay. It's a sugar that we look for. It's like like you look for other sugars, like if you're diabetic or not, people do sugar test, right? It's something similar. So insect sugar is called Trehalose. So we have the ability to look, but you have to send some live bees. We extract the hemolymph or the blood of the bee, and then we do this Trehalose assay. But that's the only way you can figure out if your bees are starving, or look at the protein levels in their hemolymph. That's another way to look at as well. But just to look at the larva, that will tell you if they're not in decent amounts of brood food floating in them, then probably you have a problem. The next question is not a question, but something for you guys, or what is it? Community answer?
I took care of that. Thanks.
Oh, okay. Oh, you did. Okay.
Since there's not a question on right now, I would love to be part of your pollen research stuff. And so I'm just going to offer to everyone who's on here, if anyone else is interested in doing it, if you could email me at vice-president@portlandurbanbeekeepers.org. I'll collect a group of people together, Ramesh, and then we can coordinate with you directly, so you don't have to.
Yeah, absolutely.
You don't have to touch base with like a whole bunch of people. We can all maybe get together at one time.
Yeah, perfect. That would be great, Jenna. Yeah. So, and then we can do a Zoom call. I can, if I don't have time, I'll ask the student Max Simon. He has done this, so he can do a Zoom call just with the participants that are interested, and he will show you other videos we have. And there is a sheet that you have to fill in if you're interested. And so there are a lot of different details that we can give you if you're interested. And it's not just this year, you can do next year too, if you miss some things this year. But yeah, we have been ambitiously looking for so many different crops and native plants, whatever is there, anything that is required for bees. We are not interested in plants or species that bees don't go. So we are only interested in the ones that bees use them as a resource for pollen.
Okay,
Yeah, that's
well,
awesome.
if anyone wants to connect in on that group, just shoot me an email in the next few days and I'll try to get a group together.
Perfect. Yeah.
Cool. And we can post on the Facebook as well, Jana, on the Facebook
Yeah.
group.
And before
Okay,
I
yeah.
forget, Ian, I think you mentioned something about swarming contacts or whatever. So I think I sent something to Rosanna, who is the Beeline editor. I don't know how many of you read the Beeline that is from the Oregon State Beekeepers Association. Maybe I'll forward that old article back. So this is because this is a swarm season, and maybe you can be a participant in another study that we did last year, but we didn't get that much data. We were late. So the question for you, how many have you read anything about swarm? I know you all know probably the basics, that half of the bees with the old queen, they go hang on to a fence or a branch or a tree or whatever, and then they look for a permanent location, right? That's what you have all understood, the swarming process. But then if you read, there is some, I think Tom Seeley's student, Juliana, who is in Texas A&M now, they did some published work in the past where they have shown, like in an observation hive, they created a swarm on an observation hive, which is not very natural. But based on that, they have said that in some cases, 70 to 75% of the bees go away with the old queen. Isn't it a disaster for the parental hive if that many bees go away? So I have been always curious about, maybe that's not true. But if you read some older papers like 60s and 70s, they all say 50% or 55, 45, something like that, not 70% bees going out in the swarm. So we did some last year. So what I want from your group is, if you see a swarm, it's not possible for all, but suppose the swarming is happening in front of your eyes, you know your hive swarmed and you know where the swarm is. If you can get a box and get all those bees back into some empty frames, so I want you to quantify the bees. So there is a protocol I can send, and I think I sent this to the Beeline last year. If you look for an issue in May or June, or I can forward that again. There are instructions on how to do. So then you come back to your parental hive and you estimate bees on both sides of the frame. So there is a way how you can estimate. I'm not looking at exactly number of bees, but you can say this is like 70% full on this side, 80% on this side, and then I can estimate the population based on that. So I have some pictures also on how to do this. So that's an easy study if you think you caught a swarm and you know which hive swarmed, right? So you go back to the hive that swarmed, and then you quantify all the bees there, and then you quantify the bees in the swarm. That tells me what percentage bees have left. So that could be a very beautiful study using citizen scientists like you guys. But I know it's not always possible. You don't know where the swarm went, or by the time you look at a swarm, you don't know which hive swarmed. And so those are different things. But if you happen to know which hive swarmed and you happen to catch the swarm, then it's an easy one. Just call me and I will tell you what to do.
That's awesome. That's super interesting. Never really thought about that.
Yeah, because there is still a debate on how many bees go. And it's a fun one. It's nothing about your beekeeping, but it's fun to involve in some basic science like that too.
Yeah, I wish I had known about that on April 17th when I looked out the kitchen window, went, oh my gosh, my bees are swarming, I gotta go.
Oh, okay. So yeah, April has been a tricky month. Yeah, I know some of you saw, but it was wet as well, right? I think this is the wettest I have seen in the last 13 years living in Oregon. So yeah, that's another study if you're involved, but that's not that priority. The priority is the pollen one, but if you're interested in the swarm one, just give me a call and I'll send you more details on what to do. But that's a basic gist. You have to quantify bees. So you give the swarm that you caught some 5 frames, and then you quantify bees on those to see how many frames of bees you have. Sometimes they may layer up in 2 layers, then it's difficult. So you have to put at least 5 frames. Then they may have some uniform layers, and you can quantify bees and tell me what population. So yeah, but I know it's not that easy, but I have more instructions in that Beeline article, but I can forward you that again.
Yeah, if you would, that'd be great.
Yeah, perfect. Okay, thank you very much. And great questions. Yeah, Portland Urban is always a great group to talk to.
Yeah, well, thank you again for taking time to talk with us, and I really appreciate it. So we'll connect with you on the pollen study and
swarm study. Yeah.
Yeah, and the swarm study too. So
perfect. Yeah.
Thank you very much, Ramesh.
Just real quick, someone just private messaged me that they were trying to email me at the vice-president email and it wasn't working. So if you want to connect with me on the pollen study, just email me to my personal one, jana.m.patterson@gmail.com.
Cool. And we can also post it on Facebook too and have people respond there as well. If that's
Yep.
easier. Yeah.
Thank you so much, Ramesh.
No problem. Take care. Good night.
Thank you. Bye, everyone.